Every plant you have ever grown is a ghost, not in a supernatural sense, but in an evolutionary one. The tomato on your windowsill carries the genetic memory of a wild ancestor that produced fruits the size of a marble, bitter enough to deter most animals, hidden in dense thorny undergrowth in the Andes Mountains. The rose in your garden is a pale imitation of a scrubby, five-petaled wild ancestor that flowered once per year and spent most of its energy on hips and thorns rather than on the saturated, perfumed blooms we have bred it toward. The sweet corn in your backyard descended from a grass called teosinte, whose grains are so hard and so few per plant that the transformation from wild ancestor to modern crop took thousands of years of deliberate human selection. The plants we grow today are edited, not in the genetic engineering sense, though some are that too, in the older, slower sense. Edited by 10,000 years of human preference, human need, human aesthetic, and the particular conditions of human cultivation. Every generation of selection pressure pushed plant characteristics in specific directions: bigger fruit, more flowers, less defense, faster growth. And with each push in those directions, other things were quietly lost. What were those things? What did ancient plants have that their modern descendants no longer possess? The answer is more interesting, more scientifically documented, and more practically relevant to your garden than you might expect. Because in many cases, the characteristics that were selected away from were not weaknesses. They were extraordinary biological adaptations that solved problems modern plants no longer solve particularly well. Before we get into the list, one clarification that matters. The word lost in this context does not mean gone from the plant kingdom entirely. Many of the characteristics described here still exist in wild plant relatives, in heritage varieties, in traditional cultivars maintained by seed-saving communities, and in the gene banks where researchers preserve plant genetic diversity for future breeding programs. What has been lost is the routine presence of these characteristics in the domesticated, commercially available plants that most gardeners work with. The loss is from the common garden, not from the planet, and that distinction matters because it means access to these characteristics is not purely theoretical. It is a question of which seeds you choose and where you source them. These are 10 of them. The first is thorns and physical defenses. Modern plants are dramatically less spiny, less thorny, and less physically defended than their wild ancestors. This sounds like an improvement, and from a human usability standpoint it often is. But the thorn was not simply an inconvenience that happened to evolve. It was a sophisticated defense system that deterred grazing animals, protected the growing points of the plant, and in some species, channeled rainwater toward the root zone by directing water along the spine toward the base. Wild roses had thorn densities that made them nearly impenetrable to large herbivores. The domesticated descendants that now grace formal gardens have thorns that are mostly vestigial, present but sparse enough to pose no real defense. Wild cucumbers were almost unhandleable by hand. Modern cucumbers are smooth. The physical defenses that protected wild plants from predators have been systematically removed by thousands of years of human preference for plants that are easier to harvest, easier to work with, and more pleasant to be near. The ecological function of thorns extended beyond simple deterrence. In many thorny plants, the architecture of the thorn placement was as important as the thorn itself. Roses positioned their thorns at angles that made downward movement through the canes easy for pollinators and birds seeking fruit, while making upward movement by ground predators difficult. Some cacti ancestors positioned their spines to create a microclimate of trapped air that reduced water loss from the plant body during intense heat. The thornless or nearly thornless modern roses and cultivated fruit trees that fill nursery catalogs are easier to manage, but they have lost not just a physical characteristic, but the entire ecological logic that thorn architecture represented. The second is chemical bitterness and toxicity. Wild plants were dramatically more chemically defended than their domesticated descendants. The bitterness that appears in modern lettuce, in heritage varieties of cucumber, and in certain apples, is a mere shadow of the chemical landscape that wild plant ancestors maintained. Wild lettuce, Lactuca virosa, produces a milky latex containing lactucarium, a compound with genuinely significant sedative properties that was used medicinally throughout the ancient world. The domestic lettuce in your salad bowl produces almost none of it. The cyanogenic glucosides in wild almonds make them genuinely toxic in significant quantities. The domesticated almond selected for sweetness contains almost none. The capsaicin in ancestral chili peppers was maintained at levels that deterred mammalian feeding, while allowing bird dispersal. because birds cannot taste capsaicin and mammals can. Modern cultivation has taken capsaicin in both directions, toward extreme heat for human culinary use and toward zero heat for sweet pepper production, but the careful ecological calibration of the wild ancestor is lost in both directions. These chemical compounds were not incidental. Many of them drove the evolution of entire insect and animal communities. The milky latex compounds in wild plants deterred certain insects while being tolerated by others, creating complex coevolutionary relationships between specific plant lineages and specific insect lineages that persist in reduced form in some wild plant populations, but are largely absent from domesticated plant chemistry. The third is deep and complex root architecture. Ancient plants and their wild relatives maintained root systems of extraordinary complexity compared to modern domesticated varieties. Wild wheat relatives produce root systems that penetrate the soil to depths of 6 feet or more, accessing water and mineral reserves far below the surface. Modern high-yield wheat varieties have shallower, more lateral root systems optimized for the uptake of surface-applied fertilizer rather than for deep mineral scavenging. Wild tomato relatives in the Andes produce root systems that penetrate volcanic soils through networks of extraordinary density, associating with mycorrhizal fungi across a much wider zone than their domesticated descendants. The selection for above-ground productivity in virtually every crop species has come at the cost of below-ground investment. The roots got shallower and simpler because the soil got richer with human inputs. A plant that does not need to search for nutrients does not invest in the root architecture to search for them, and in that shift, generations of wild root complexity were quietly lost. The fourth is night flowering and nocturnal pollination. Many wild plant ancestors were adapted for pollination by moths, bats, and nocturnal insects. They opened their flowers at dusk, produced intense fragrance that was specifically tuned to carry on night air, and in some cases reflected ultraviolet light in ways that were visible to moth-compound eyes. Modern cultivation has shifted plant flowering almost entirely toward day-blooming varieties, because daytime flowering is visible to human observers, is more compatible with human working schedules for manual pollination and harvest, and is pollinated by honeybees whose economic importance in commercial agriculture has driven the entire system toward bee-compatible timing. Tobacco is one of the few widely grown plants that still shows significant remnant night flowering adaptation, with wild tobacco relatives producing strongly night-fragrant flowers specifically tuned for hawk moth pollination. Most of the crop plants that once featured this adaptation have lost it entirely, and with it, the nighttime gardens that their wild ancestors created, complex aromatic environments that supported entire nocturnal pollinator communities, are essentially gone from the domesticated plant world. The fifth is mycorrhizal dependency and soil relationship. Wild plants exist in profound chemical conversation with the soil around them. That conversation is conducted primarily through the mycorrhizal fungi that colonize root systems, forming connections that allow the plant to access phosphorus, trace minerals, and even water from far beyond the reach of its own root tips. Wild plant species that evolved in poor soils developed mycorrhizal associations of extraordinary specificity and effectiveness. Modern domesticated plants grown for generations in intensively managed fertilized soils have progressively reduced the sophistication of their mycorrhizal relationships. High phosphorus fertilization specifically suppresses mycorrhizal development because the plant does not invest in maintaining a biological relationship to access something it is already receiving in abundance. Over generations of cultivation in high phosphorus soils, the genetic capacity for robust mycorrhizal association has weakened in many domesticated plant lineages. A wild plant transplanted to poor soil can often establish and thrive through its mycorrhizal network. The equivalent domesticated variety frequently cannot, because the biological capacity for that relationship has been diminished by generations of selection in conditions where it was not needed. The practical implications of reduced mycorrhizal capacity in domesticated plants are beginning to be felt more broadly, as agricultural systems increasingly seek to reduce synthetic fertilizer dependence. When modern crop varieties are planted in soils without high phosphorus inputs and without synthetic fertilizer supplementation, they frequently underperform relative to expectations in ways that their wild counterparts, growing in similarly poor conditions, do not. The difference is often attributed to soil poverty, but a growing body of agricultural research suggests that a significant portion of the performance gap is due to the reduced mycorrhizal networking capacity of the domesticated varieties themselves, which were bred under conditions that made the mycorrhizal relationship unnecessary and allowed it to deteriorate across generations of selection. Restoring that capacity is now an active area of plant breeding research, essentially attempting to reintroduce a biological relationship that was inadvertently selected out. The sixth is allelopathic chemical communication. Ancient plants communicated with their competitors through chemical signaling in ways that modern domesticated plants do mostly inadequate. Allelopathy, the production of chemical compounds that suppress the germination and growth of competing plants in the surrounding soil, was a primary competitive strategy in dense natural plant communities. Wild black walnut remains one of the most dramatic examples still visible in nature, producing juglone, a compound so effective at suppressing competitors that the zone beneath a mature black walnut is often noticeably sparse compared to surrounding vegetation. Wild sunflower relatives produce allelopathic compounds that clear space around them in natural prairie ecosystems. Many wild herbs produce volatile allelopathic compounds that function above the soil surface, chemically deterring competitors from establishing near them. These systems were selected away from in domesticated varieties, partly because they could affect neighboring crop plants in mixed agricultural systems, and partly because they were simply not visible characteristics that human breeders were paying attention to. The result is that modern crop plants are dramatically less capable of competing with weeds than their wild ancestors were, creating the dependence on synthetic herbicides that characterizes modern agriculture. The seventh is epigenetic plasticity, the ability to adjust gene expression rapidly in response to environmental conditions without changing the underlying DNA. Ancient plants, evolving in highly variable environments, developed extraordinary capacity for epigenetic adjustment. The same plant could express dramatically different phenotypes, different leaf shapes, different root depths, different chemical profiles, in response to drought stress, temperature change, or nutrient availability. Modern domesticated plants have been selected for consistency and predictability. Growers need plants that perform the same way under the same conditions across large areas. The variability that epigenetic plasticity produces is a disadvantage in an agricultural system that wants uniform performance. And so, generation by generation, the high plasticity genetic capacity of wild plant ancestors has been progressively narrowed toward predictable, consistent expression of a narrower range of traits. The plant became more controllable and less resilient at the same time. The eighth is secondary metabolite diversity. Wild plants produced extraordinary arrays of secondary metabolites, chemical compounds with no direct role in basic plant growth, but with important functions in ecological interaction. Flavonoids, terpenes, alkaloids, phenolic compounds, compounds with names that require a chemistry background to fully appreciate, existed in wild plant populations in diversity and complexity that modern domesticated varieties do not approach. These compounds are now understood to function as UV protection, as insect deterrents, as disease resistance compounds, as signaling molecules in plant-to-plant communication, and as the primary drivers of the nutritional complexity that characterizes truly wild-grown food plants. Heritage variety tomatoes, selected for flavor rather than yield, contain measurably more lycopene, more flavonoids, and more diverse phenolic compounds than modern commercial varieties. Wild bilberry contains higher anthocyanin concentrations than commercially cultivated blueberries. The nutritional gap between wild and domesticated food plants is increasingly documented in nutritional research, and it reflects the loss of secondary metabolite diversity that accompanied the selection for yield, shelf life, and appearance. The ninth is dormancy complexity and environmental response. Wild seeds from ancient plant lineages exhibited dormancy systems of extraordinary sophistication. Some seeds required specific temperature sequences to germinate, cold followed by warm followed by a further cold period, because they had evolved in environments where the wrong germination timing was fatal. Some required the specific chemistry of smoke from natural fires, having evolved in fire-adapted ecosystems where germination cued to fire meant germinating into a suddenly cleared and nutrient-rich landscape. Some required physical scarification by the digestive systems of specific animals that carried them, germinating only after passing through a gut that both softened the seed coat and deposited the seed in a nitrogen-rich location. Modern agricultural seeds have been selected away from complex dormancy requirements because agricultural systems need reliable, predictable, rapid germination under standard conditions. The extraordinary environmental intelligence of wild seed dormancy has been largely removed from domesticated plant genetics in favor of seeds that germinate whenever you plant them. The smoke germination response is particularly striking as an example of evolutionary specificity. Characins, the chemical compounds in smoke that trigger germination in fire-adapted plants, were only identified and named by researchers in the early 2000s, though traditional farmers in fire-adapted ecosystems had used smoke to stimulate germination of certain seeds for centuries without understanding the chemistry. The discovery that a class of compounds produced by the combustion of plant material had been so specifically incorporated into the germination biology of entire plant families over millions of years of coevolution with fire is a reminder of how sophisticated the biological systems that were quietly removed from domesticated plant genetics actually were. Modern agricultural seeds that germinate reliably when planted at a consistent depth in moist soil have exchanged that sophistication for compliance. Both have their value. The difference is that wild dormancy complexity cannot yet be engineered back in. It can only be accessed through the wild relatives and heritage varieties that still carry it. The 10th is cooperative root signaling. Recent research in plant biology has documented that plants in natural communities engage in sophisticated underground signaling through mycorrhizal networks, root exudates, and volatile compounds. Wild plants in established communities send chemical signals that warn neighboring plants of insect attack, that share photosynthate through fungal networks with shaded seedlings of the same species, and that respond to signals from root competitors in ways that alter their own root growth patterns. This underground cooperative and competitive signaling network, sometimes called the Wood Wide Web in popular science writing, is dramatically reduced in domesticated plant varieties grown in agricultural settings. The combination of simplified monoculture planting, high fertilization that reduces mycorrhizal dependency, and the selection for individual plant performance rather than community behavior has progressively isolated domesticated plants from the signaling networks that their wild ancestors participated in as members of complex ecological communities. What does any of this mean for the person standing in a garden or a kitchen, with seeds in their hand and dirt on their gloves? It means something more hopeful than it might initially sound, because many of the characteristics that were selected away from in domesticated plant varieties still exist in the genetic material of wild relatives, heritage varieties, and traditional cultivars that were never subjected to the full force of commercial selection pressure. The wild tomato species in the gene banks of South America still carry the deep root architecture. The heritage wheat varieties that were nearly abandoned when high-yield dwarf varieties took over in the 20th century still carry some of the mycorrhizal association capacity. The old-fashioned, open-pollinated vegetable varieties that were replaced by hybrid seeds in the mid-20th century still carry higher secondary metabolite diversity than the hybrids that succeeded them. Growing heritage varieties is not nostalgia. It is access to a biological legacy that commercial plant breeding has not yet found a way to fully package and sell. The flavor is different. The resilience is different. The relationship with the soil around them is different. These are not subjective impressions. They are measurable differences in plant chemistry, root architecture, and ecological behavior that thousands of years of human cultivation edited away, and that exist now only in the varieties that were overlooked, preserved, or never domesticated in the first place. There is a growing movement among plant scientists, seed savers, and farmers to recover some of what was lost. Perennial grain programs are attempting to breed the deep root architecture of wild grass relatives back into productive grain crops. Participatory plant breeding programs are working with traditional farming communities to document and maintain cultivar diversity that commercial breeding overlooked. Botanical garden seed banks are preserving wild plant relatives that carry characteristics their domesticated descendants no longer possess. These efforts represent a recognition that the 10,000 years of selection that gave us the productive, beautiful, manageable plants in our gardens and our grocery stores also made choices that we are only now beginning to fully understand, and in some cases, to regret. The plant in your hand right now is the product of an extraordinarily long human conversation about what a plant should be. That conversation is not over. Every choice you make in a seed catalog, every heritage variety you grow, every wild plant you leave standing at the edge of your property, is a contribution to the next chapter of that conversation. Drop a comment telling me which of these 10 lost characteristics surprised you most, and whether you grow any heritage varieties in your garden right now. I read every comment. Give this a thumbs up if it changed how you think about the plants you grow. And subscribe for plant science content that goes deeper than the garden center label every single week.